So in this lecture, I'm going to talk about neural physiology, okay, like how a neuron actually work. So we talked about, like, we know that nerve creates signals. So what does that signal means? That signal means production of action potential. That is exactly what we're going to talk about. Okay, so let me get the PowerPoint. So neuron, like skeletal muscle, because we have some cells in our body. So neuron is one of them. Neuron, like skeletal muscle cell, can be excited, meaning they have excitable plasma membrane. So their membrane, we can make them excited. That's what it's called, electrically excitable membrane. Now, why? Because they do have something called resting membrane potential. Like, what does that mean? That means is this. So let's say this is a membrane. This is the plasma membrane right here. So inside the cell, that is outside the cell. So this is interstitial fluid or extracellular fluid. This is intercellular fluid, okay, or cytosol. So inside the cell, we have multiple types of ions. Like it could be like sodium, potassium, chloride, phosphate, protein, different type of ionic environment. Outside the cell, same is true. Now their distribution is not same. So some ions outside more, some ion inside more. In this chapter, we are going to focus on the two ions. Those two ions are one sodium ion and the other one is potassium ion. These are the two ions we are going to focus on. So now they move. How they move? The question is how ions move. We learned that ions cannot move through the plasma membrane because inside the plasma membrane we have hydrophobic core. So ion, being an ion, cannot, they have charge, so they can, whether positive or negative, they cannot move through those hydrophobic cores. But they have ion channels. Through those channels, they can move. Some ion channels, they can move passively, meaning those ion channels are happen to be like so-called leaked channels. In this picture, this is a leak channel. This is another leak channel. So potassium and sodium, they are moving through those leak channels. Obviously, they are not that much. So that many we don't have. So obviously, that's not the real way of ion movement. What is the main way of ion movement? Those are called gated channels. So those are regulated channels, meaning cell will open at a different circumstances. We're going to talk that later on. So now coming back here, when this, because of these leak channels, all these ions move in multiple directions. Like they can, some ions move from inside to outside, some move from outside to inside based on different criteria. And as a result, when they reach at the equilibrium, meaning they are not, after that, they are not significantly moving, when we measured the voltage, when we measured the differences of their net charges in both sides, we find that's negative 70 millivolt. That is why it is said to be that resting membrane potential for neuron is negative 70 millivolt. Resting membrane potential is negative 70 millivolt. Now, what does that mean? The answer is in the statement. Resting membrane potential, meaning when the cell is at rest, that is the potential. Now, what is negative means? Very important to understand. Negative in this case means inside we have more negatively charged ion. So inside is more negative environment. That's what these lines are for. The inside are more negative environment compared to the outside. This does not mean that outside we do not have negative charge ions. It just means inside we have more negative environment. Okay? That negative side indicate that. And 70 is the numerical value and millivolt is the unit in this case. So do not forget this, that resting membrane potential in case of neuron cell is at negative 70 millivolt. Now we're going to say, like, how it can change. So in order to understand, let's focus again on those two ions. One is sodium, another is potassium. So pay attention to this particular picture. Let's say that we allow potassium to move. So we allow potassium to move through those leak channels. Now, question is which way they will move. So movement of ions, actually, they are governed by two properties. One is their chemical, electrochemical gradient, meaning in short, no, not electrochemical gradient. One is chemical gradient, another is electrical gradient. Together, we call electrochemical gradient. Now, what is chemical gradient? That's based on their concentration. And what is electrical gradient? That's based on their charge. So we already learned that inside, we have a little bit more negatively environment. So according to the charge, potassium ion, they want to move inside because potassium ion is a positively charged ion. Inside is a negatively charged environment. We know opposite attracts. So any potassium ion in the vicinity, their tendency is to go inside. That's one. The second part is, this is a fact that I'm going to give it to you, that inside, in the cytosol, we have greater concentration of potassium compared to the outside. On the other hand, in the interstitial fluid, means outside, we have greater concentration of sodium. So these are the two facts we have to remember. So inside, in the cytosol, we have greater concentration of potassium. So potassium concentration is higher. Outside, sodium concentration is higher. So for sodium, from outside to inside, that's a gradient. For potassium, inside to outside, that's a gradient. So if we follow that, now look at the picture again. So right here, potassium, according to the chemical gradient, meaning concentration gradient, their inside is very high, outside is less, so they want to move outside. According to their electrical gradient, they want to come in. So they are having two opposing forces. So one force is telling them to go out, the other force telling them to come in. So it's like in your head, like one force telling you to study hard so that you can be very successful according to your grade and other thing. And the other force say, oh, I just don't want to study, I want to watch a movie. So it's like two opposing forces. Depending on which one wins, you will do that. Same thing here. When scientists did the experiment, they found that chemical gradient, concentration gradient, in this case for potassium, wins a little. That's what this narrow arrowhead means, meaning together the net tendency is to go out. So potassium, together the net tendency is to go out. That's very important to know. Now let's look at sodium. Sodium, according to their chemical gradient, sodium, according to this is the sodium, according to their chemical gradient, they want to come inside because outside is a higher concentration compared to inside, so sodium wants to come in. According to their electrical condition, they also want to come in because they are positively charged and inside is negatively environment. So together, if you put them together, their net is to go inside. Their net tendency is to go inside. So in summary, potassium, their net tendency, although weak, but still want to go outside. And sodium, they have a tremendous tendency to come inside the cell. That's the summary, okay? But that doesn't mean they can't move because they are ions. So we know ions can only move mostly through the gated channels. If we allow them to freely move, what would happen? So if we allow potassium to freely move, this is what would happen. The electrical equilibrium potential would be negative 90 millivolt. On the other hand, if we freely allow sodium to move it would be positive 66 millivolt. That would be the plasma membrane potential, but that is not the case because they can't do this. They can only be up to negative 70 millivolt after that weak tendency and the greater tendency when they settle down. So in other words, The negative 70 millivolt is the plasma membrane of the neuron cells resting membrane potential. And we also learned potassium has a weak tendency to go out of the cell whenever the opportunity comes. And sodium, they have a tendency to go in a great tendency to rush into the cell whenever the opportunity comes. So now we're going to talk about those opportunities. So what happened, as I said earlier, that there are two types of channel mainly. One is called leak channels and one is called gated channels. So leak channels already justified our negative 70 millivolt resting membrane potential. We already know that. So what are the gated channels? There are three variety of gated channels. One is called voltage gated channel. The other one is called ligand or chemically regulated gated channel. And the third one is called mechanically regulated gated channel. So what does that mean? So as the name suggests, if the voltage of the membrane changes, so right now the resting voltage is negative 70. If that changes, whatever the change is, we're going to talk about that later. If the changes significantly, then there will be some gate which will open up. It's like electrical gate, like you go into some gates you have seen in multiple offices and colleges building, like you go close to that gate, they have a motion sensor, they sense you and they just open automatically. Or there are other type of gate, you literally push the handle and then you open it forcefully. That's a mechanical gate, okay? So in this case, mechanical gate means, for example, if I touch something, so when I'm touching, let's say I'm touching this, so my cells are mechanically distorted, so they are opening up some gates. And chemical gates means when hormone or neurotransmitters, such as you learned this in neuromuscular junction in the skeletal muscle chapter, that acetylcholine come outside the synaptic knob and bind to the receptor. That is a chemical. So when that chemical bind, gate can open. Those gates are called chemically regulated gate. So we have three types of gate. If the voltage changes, gates can open. So in response to the voltage, gates can open or close, in other words. That's a voltage regulated, similarly chemically regulated, mechanically regulated. Okay. So now we will learn like what happened when a chemical comes or what happened when a mechanical changes happen. So in this particular example, if you look at this picture right here, this is the gate. This is a chemically regulated gate. When these chemicals are coming to attach, the gates open up. That's just a description like how it works. This is a voltage gate. So when the voltage is minus 70, like negative 70 millivolt, when it becomes negative 50, for example, this gate will operate. So the gate will allow molecules, ions to move in or out. So this is the example of those kind of gates. So let's say on a neuron membrane, in some place, there are some chemicals or a mechanical changes happen. So maybe acetylcholine came or any other neurotransmitters came, or you mechanically touch something, so those changes happen, either or. So what will happen? So the gate will open up at that area. So let's say this is the membrane. Let's say right here there is a gate and some chemical changes happen, so this gate will open up. Now, just focus yourself. Let's just think that let's say we are talking only sodium gate. Forget about anything else. Only sodium gate. So sodium gate will open up. Now we already learned that sodium has a tremendous tendency to move in. Sodium has a tremendous tendency to move in because they are electrical and chemical, both gradients telling them to move in. So they are just here. Tons of sodium are sitting here. They just can't move because they're waiting for gate to open. So it's like you went to a concert, so let's say thousands of people outside the gate to just go in. So they're just waiting because gates are not open. As soon as the gate open, they will rush because that's their tendency. It's like buying iPhone. So you're at the Apple store, you're waiting online 2 days, okay, so because you want to go in, but you can't because the gates are closed. So as soon as mechanically somebody opens up, we rush into that store. Same thing. Sodium are waiting, they can go in because the gates are closed. As soon as some chemical changes happen or mechanical changes happen, those gate of sodium opens up, they will go in. Now the question is, what will happen to that voltage? Remember, the voltage was negative 70 millivolt. As soon, negative 70, pay attention to this one. Voltage is negative 70 millivolt at rest. Now sodium gets in. Just think numerically. So let's say one sodium gets in. So sodium is a positively charged ion. As soon as they get in, the voltage will change. Which direction? They will change to negative 69 because it's a positively charged ion. So when the positively charged ion goes inside, the negative environment reduces. So they are going to move from negative 70 to negative 69. So what I did, this is the part where I like to write down, but unfortunately this whiteboard does not allow me to write down so nicely. So what I'm going to do, I'm going to move this camera towards my paper here. So I'm going to try to explain something else. So hopefully you'll be able to see that a little bit better. Okay. So hopefully that you'll be able to see that a little bit better. Okay. Okay. So this is the membrane. This is the membrane. At rest, it is negative 70 millivolt. And this is called resting membrane potential. That is at rest. So everywhere the voltage is negative 70. So let's say some chemical activity or mechanical activity takes place right here. So as soon as this chemical or mechanical activity takes place right here, the sodium ion will come in. So when the sodium comes in, this particular area becomes negative 69 because there is no effect down the line. Then let's say more sodium comes in, so a little bit more, the gate is open for a little bit longer. Just assume that. So initially when the concert get open, one person go in, then 2, then 3, then 4, then 30. Same thing here, the sodium coming in. So what sodium coming in, what will happen? This will be negative 68, but then this place will be negative 69 because this is more sodium coming and they're walking through the wall. It is exactly like when you walk into a concert hall. You go inside and you just go not in the middle, you normally go inside and then you kind of walk and go through your place. Same thing here, just imagine you have all the negative ions sitting on the membrane. So sodium being a positive ion, they're going to walk through the wall. That phenomena is sometimes called local current. Because of that local current, the potential is slowly changing. So it becomes negative 68, negative 69. Then it becomes negative 67, negative 68, negative 69. But this is unaffected. That means this potential, this type of potential, which is affecting a local area only, but they're diminishing along with the distances. So more you go, they have no effect. So this side, they have not effect at all. Only here, they have effect. So this type of potential we call graded potential, graded potential. Graded potential, meaning they are diminishing or decreasing along with their distance. But now imagine if they keep coming and they can reach at this particular voltage, like it will reach. So it's like, again, the concert hall, example, like let's say they open one gate and 100 people walked in. Then suddenly the manager of the concert hall will realize, hey, it's too many people, one gate is not enough. He will instruct, hey, open the next gate. So that's exactly the time. So when they keep coming, when they reach at negative 55, remember we talked about that? There are other set of gates called voltage regulated gate. They will open up. This particular potential is known as threshold potential. Threshold potential, meaning at that potential, the voltage gets open up. At that potential, the voltage get open up. Now, more get open up right here, right adjacent to it, because that reaches negative 75, so negative 55. So more sodium will come in. So obviously this place will reach negative 55 instantaneously. New gate will open up. Now more sodium will come in. Then the next will be 55 because more sodium coming in. So they will coming in and more sodium will come in and so on. So now you cannot stop. It's like dominoes effect. Once you successfully push one domino to the second one, and that's it. You don't have to do the rest, because then the second one will drop on the third one will fall on the fourth one, and so on. Same thing here. So more sodium coming in changes this potential to negative 55. New gate open up because those are voltage gate. More sodium come in from that gate. This changes to negative 55. Then more sodium come in, and the system will continue doing that until that potential, that changes reach at the end. This is known as action potential. Action potential, that is the signal. That is how a neural cell carry a signal. So let's come back here. So that is how a neural cell carry a signal. So first, one particular local area, they will open up. Meaning it's like you are pressing, again, think about, you are just pushing the first dominoes. It didn't fall, but you're putting enough pressure. Now, depending on how heavy the domino is, if you put enough pressure, they will fall. Once they fall, that's the threshold, meaning you reach the threshold. You cross the threshold, that's it. You don't have to do anything else, because then next, the voltage gate open up, they keep doing the rest. That is called action potential, because that does not decreases, that does not diminishes along with the distance. So graded potential will decrease. So if you touch the domino enough force that it does not fall, that means the domino effect you will not see. You can only see if you can make it fall. If the first one does not fall, that means you did not reach to the threshold. That means that neuron is not going to put out the action potential. The action potential can only happen when that first domino falls. Okay. So now two phenomena we got to know. One is two names I'm going to introduce. One is when the potential is moving from negative 70 to negative 69 to negative 68 to negative 67 towards zero, we call it. So when they're moving towards that direction, they can go up to positive 30 until then, because until then, the membrane is not reacting. They can tolerate that change. It's like concert hall. People are going in, but let's say 1,000 people went in. Then the concert hall has no room. They said, hey, shut down all the doors. We have too many people. We can only handle 900, so let some goes out. That's exactly happened here in neuron cell. So neuron, so many sodium came in, it can go up to positive 30. So when the movement goes that way, we call the cell is depolarizing. Depolarizing. So normally it is negative 70 millivolt. When it moved towards, it does not, like negative 70 does not has to reach always positive 30. When it goes towards that, we call it depolarizing. So a cell is depolarizing means the sodium or positive charge ion is coming in and their membrane potential is moving towards zero. Again, the cell locked their membrane at 30 because they reached at the highest point. They cannot tolerate any more changes. It's like the concert hall example that I gave you. Then what they do, they send some positive ion outside. Because remember, positive ion came inside to change that. Now we want to repolarize the membrane, repolarize. That means when the membrane potential come back from positive 30 towards negative 70. That direction is called repolarization. And how can we achieve that? We can throw those positive ion outside. Now, which one we want to throw? Cell is intelligent because remember, potassium, they are also positive and they want to go. So some people, let's say 1,000 people came in the concert, not everybody likes Taylor Swift or whatever you like. Some people don't like it. They say, hey, I don't know that this is the singer, so I'm going to leave. That's the potassium. So potassium wants to leave. And cells say, hey, open some potassium gate, let them leave. So when potassium leaves, then the membrane comes back to the normal negative 70 again. So the cell depolarize and then the cell repolarize. That's what the action potential curve look like. So let's look at the curve. Okay. This is what we already discussed about all the resting potential. You can read from this particular slide about the liquid membrane. These are all the mechanical channels and gated channels that I talked about using the paper. So hopefully you will understand that in paper. This is the graph I'm talking about. So right here, it is negative 70 millivolt. Right here, this is negative 70 millivolt. Now the stimulation came, meaning the chemical or mechanically regulated gates open up. the membrane potential changing slowly, changing slowly. This is graded potential, graded potential. Once it reaches to the threshold, once it reaches to the threshold potential, new gates open up, voltage gate open up. So that voltage gate will take boom to the positive 30. That is depolarizing phase. That means the cell is depolarizing. And then once they reach there, they will close all the gates. Now, who is going out? Potassium is going out. When potassium goes out, because that's also a positive ions, so they keep decreasing. That's called a repolarizing phase. And they keep coming to negative 70. But what happened is, it's like closing the gate. So when it reaches to the negative 69, cell is saying, hey, close the gate. We don't want to go beyond negative 70, close the gate. But before they close the gate, just imagine that, few more potassium goes out because their tendency is to go out. They want to go out. So once few more goes out, the potential dip a little bit. That's called hyperpolarizing. So that's called hyperpolarizing phase. Like they hyperpolarized, but because of the leak channels, the equilibrium comes back, so they come back to the resting potential. So this is what action potential is. So neuron membrane can produce the signal through this mechanism. So this is just telling you about all the ions. So basically at the resting stage, when you open up, the sodium ion comes in, potassium, we are not doing anything. Depolarizing state, more sodium comes in, the threshold potential become negative 55, then it goes up to 30, then they close the sodium channel, but they now open up potassium channel. Now potassium is going back, bringing the potential back to the negative 670 after hyperpolarization is adjusted. That's basically, so depolarization is changes from negative 70 to 30, and repolarization is the reversal of that. This is explaining how sodium and potassium is functioning. So sodium, they allow the membrane to come, they allow the ions to come in. So again, that is depolarization. This is initially the graded potential, then the voltage gate, so the action potential. And then once they reach on the top, now the sodium is closed, potassium open up, so new molecule potassium goes out, so the repolarize complete. And that's so 1, 2, 3, and 4. That's how it works. So slowly potassium is sending outside. Now potassium is leak and sodium leak channels adjusted to make it negative 70. Okay. This is exactly the same thing, but I'm going to mention something else. That's a new word. So basically we know this. We know this part because negative 70 is the resting membrane potential. One more time. This is graded potential, negative 55, that threshold. At the threshold, voltage gates open up, they go up, depolarize, and then they repolarize. They should be stopping here, but as I explained, they can't stop because the gates are closing, hyperpolarize, then leak channel, they'll keep them back again at resting membrane potential. Now, what is important to understand here is something called a refractory period. Like, what does that mean? That means once, think about, forget about action potential. Once the dominoes are falling, you cannot make a second dominoes effect. Because remember, once the dominoes are falling, you have to put those dominoes back again and keep it nicely positioned, and then the second effect can be happen. Same thing here. Once the neuron is excited, after threshold, you can't stop it. You cannot start another one. Let them finish, then only you can do the second one. That's called refractory period. Only time you can do a second one, like you can activate you can stimulate them again if you reach at this position, meaning when they're already done, you can have a higher stimulus that can cause action potential. Okay, so what does that mean? That means a period of time when neuron cannot generate another action potential. That's called absolute refractory period, because they are not going to. So even if you send a strong stimulus, it's not going to begin another action potential. Why? Because they're already undergoing an effect. I'll give another example to understand. You know the cistern that has water back of the toilet. So once you use the toilet, if you flush the toilet, if you flush it, all the water is coming out. Now you cannot flush it twice. You cannot do it second time while the water is going. You have to wait until that is done. The new water fill the tank up, then you can do that. That's exactly like that. So once neuron is excited, let them complete their action potential, and then you can trigger the second one. Okay, so this is called absolute refractory, means you cannot really do anything. Relative means almost when the tank is filling. Remember in the toilet, when the tank is filling, almost when they're at the end, like say 70%, 80% filled, if you press, it will happen. The reaction will, the effect will, you can see. That's called relative refractory period. Like you don't have to wait at the end when the water stops pouring into the cistern or pouring into the tank. You can do it like when the 10-20% remaining. That period is called relative refractory period. Means in this case, like when they're going up there, coming down, you can't do anything. You cannot do anything. That's absolute. When they're hyperpolarized, that particular area, remember the potassium channels are closed, they're hyperpolarizing, that time a stronger stimulus can make them go up again. That's action potential. Now, we talked about that action potential, which is moving. So it takes place near the axon hillock normally, where the cell body is, and then they move towards the telodendria, where the synaptic terminal is. So when the action potential reaches to the synaptic terminal or synaptic knob, they release the neurotransmitters. We learn in NMJ, neuromuscular junction, when the action potential comes at the end, the neuron that near the skeletal muscle, they're going to release acetylcholine. There could be many other neurotransmitters. We're going to talk about them slightly later. So right now, to understand this, so the action potential is the signal comes to the new synaptic knob, and then they release neurotransmitter. That neurotransmitter going to bind with the receptor and exert their effect on the postsynaptic cell. Now, this movement in the presynaptic cell, that action potential can be two types. One is called continuous, the other one is called saltatory. One is called continuous, the other one is called saltatory. What does that mean? One is like step by step, and one is like galloping through the nodes of RANVA. Remember, nodes of RANVA, we talked about that in my previous lecture, that nodes of RANVA is a gap between two Schwann cells, so they just gallop on those areas. It is the best example to understand is like, let's say we talk about a train. So we are going, let's say from New York to Washington. Imagine a train that stops at New York, and the next stop is like somewhere in New Jersey, like Trenton, New Jersey, and the third stop somewhere in Baltimore, and the fourth stop is Washington, DC. That's like saltatory. And imagine another train going, starting from New York, they stop at next another station, let's say in New York, and then they stop at the first station in New Jersey, near the Jersey City. Then they have, let's say, 20 different stops before they move to Trenton. Then there is another 30 different stops before they move in Baltimore, and so on. So one is local train, another is galloping or express train. Continuous is the local train, which are basically unmyelinated neurons. So they don't have any nodes of RANVA, so they don't have to jump. And saltatory is like a galloping, meaning like a horse. So they move like an express train. So obviously, if the neuron is myelinated, they are going to take the signal much faster than unmyelinated neuron. So propagation means action potential when they produce two types. One is continuous, the other one is called saltatory. Now, there are three different type of fibers exist, which it's the best way to understand is let's imagine there are three different type of road exist. One is called expressway, one is called, let's say, boulevard, the other one is called street, like a local street. So expressway, you can drive like 60 miles per hour. Boulevard, you can drive like 40 miles per hour. And street, you can drive like 25 miles per hour. Same as here. So this is expressway, A, this is the boulevard, B for boulevard, and C for street. Okay, so A means expressway, so that's their fibers are the largest, and they can take really fast. They can go 130 meters per second. You don't have to memorize the number just to understand it. So these are myelinated somatic, sensory, and motor neuron to skeletal muscle. They can take really fast. Excuse me. Type B, they are, as I said, in the middle, boulevard, so they are basically myelinated, but they're sensory and autonomic preganglionic fibers. They can take approximately like 15 meters per second. Again, do not have to memorize, just remember they're in the middle. And these are the smallest fiber. So they're the smallest fiber, they're unmyelinated, and they normally are the slowest. So this is how, so you memorize type A fiber is the fastest and they're largest. Type B fiber the medium size, they're the medium speed. Type C fiber the smallest, they're the slowest speed. Now when they take, how they take, like from one end to another end. So let's say one neuron take the signal, then what they do? They give it to another neuron. That junction is called synapse. That's what we're going to talk about. Now, how can they give it? There are two types of synapses. One is called electrical synapse. One is called chemical synapse. Electrical is this, like they're connected, they're physically connected, like an electrical wire. And chemical is like Bluetooth. So they are not physically connected. So like a Bluetooth connection, like a Wi-Fi connection. So they have a gap between two. So when there is no gap, let's say they are connected directly, oh, I don't have a picture from that. That's called the electrical synapse. So that means they are faster and they are two-way transmission and they're connected like this. Their cells are connected through gap junction. But we have very few neural connection like that. We have some in the brain. Chemical one, that's the important one. That's a Bluetooth one is the important one. So they have a junction right here. That's a Bluetooth area, meaning that's a no connection area. So this signal goes here, they release it exactly like neuromuscular junction, and they releases neurotransmitter, they go and affect that. So this is called presynaptic, that's called postsynaptic cell. This is presynaptic, that's called postsynaptic cell. Now the question then, is there just a one type of flow or there are multiple types? The answer is there are multiple type of flow. Very easy to understand from the picture. So when these are the three types, axodendritic, its actually answer is in the word. Axodendritic, meaning from axon to dendron, another neuron. Axosomatic, axon to soma in the other neuron. Axoaxonic, axon to another axon. So this is the pre, that's post. So look at this. This is the presynaptic and that's the postsynaptic. So axon comes here giving to the dendrite of the postsynaptic. That's axodendritic. Then axon comes here. This is the axon. They're bringing in, this is the presynaptic cell. They're giving to the axon of the other one. This is the postsynaptic. That's called axoaxonic. Again, this is another presynaptic. They're bringing in directly to the soma. Remember, soma is the neural cell body. That's a soma. So that's called axosomatic. So chemical synapses can be three types based on, this is very important. Chemical synapses based on how they are attaching to each other. So how the junction coming, is the Bluetooth area is axon and dendron, or axon to axon, or axon to soma. Based on that, there are three types. But chemical synapses could be multiple types again, based on what type of neurotransmitter they release. Remember, their connection, their Bluetooth is here is the neurotransmitters. So they are releasing some neurotransmitter, they're getting it. They're releasing some neurotransmitter, they're getting it. They're releasing some neurotransmitter here, they're getting it. So what kind of neurotransmitter they release? So there are a variety of neurotransmitters. This I'm going to come back 2 seconds later. There are a variety of neurotransmitters. So what I did, I just put in this particular slide, the main important one that you need to know. Your book may have tons of them. You do not have to memorize them. You just need to know this one. Okay, your book has again a lot more than this. You do not need to memorize that table, or if you have a table in your book or not, you need to know this. So this you need to read a couple of times to memorize it. Acetylcholine, the one that we already know, that's in the NMJ, neuromuscular junction. That's at the skeletal muscle. I'm going to explain this word 2 seconds later. And there are amino acids, like a GABA aminobutyric acid is a neurotransmitters. Biogenic amines, some of the biogenic amines like tryptophan, they modified of them, they can be act as a neurotransmitter. Those are norepinephrine, dopamine, serotonin. These are all neurotransmitters, and also nitric oxide, NO. It's not no, it's nitric oxide. NO is a gas that can be also a neurotransmitter. These are different type of neurotransmitter. Some neurotransmitters, this is interesting. Some neurotransmitters, when they release, they can be excitatory. Okay, and some neurotransmitters, they can be inhibitory. A little bit more, this is not so straightforward, a little bit more complicated. Let me explain again. Some neurotransmitters, they can be excitatory in some case, inhibitory in other case. Some neurotransmitters, they can be mostly inhibitory. Some neurotransmitters, they can be mostly excitatory. So without memorizing those, first we need to know what is excitatory and inhibitory means. This is what it means. When the presynaptic cell releases neurotransmitters, the postsynaptic cell which is receiving it, if they receive and after they receiving, they continue that signal, that means they're excited. That's excitatory neurotransmitters. If a presynaptic neuron releases a neurotransmitter after receiving, the second neuron, meaning in this case second cell or the postsynaptic cell, does not make any big changes, that's inhibitory, meaning the signal dies right there. So it's like how happy you are. So it depends on what is your receptors. So for example, I'll give a simple example to understand this first. So let's say I, when my son is in high school, I tell him, hey, study well. And if you do study well, get 100 in the bio exam, and I will give you something. Go figure out something, okay? So let's say, hypothetically speaking, he studied well, he got 100 in the bio exam. He came back to me and said, hey, Dad, I did as you said, so where's my gift? Okay, I take out my wallet, here's the dollar. Now, he's inhibited most of the time. Most of the cases I have seen, like, oh, so he probably will go across the room, slam the door, normal things will happen. That means he was in his head, probably he was expecting like a $50 video games. But as soon as I gave him $1, that means it is inhibitory for him. But think, if I get from a train and I give a $1 to a homeless person, unfortunately, on the street, and I buy a coffee for that person, that made his or her day. It's not the dollar, it's the person who's receiving get either excited or inhibited. Same dollar can make somebody sad, can make somebody happy, because it's in them. So the receptor, when it's received the neurotransmitter, they can be either excited or inhibited. That's what excitatory and inhibitory means. So when they get excited, that postsynaptic potential, we call that EPSP, excitatory postsynaptic potential. That means the postsynaptic cell is excited. When they're inhibited, that potential is called IPSP. That means the postsynaptic cell is inhibited. So this is like in my son's case, that's the homeless person's case. That neurotransmitter can be excitatory, can be inhibitory. Okay. Now, this is an example given just to explain one more time. You studied this in the neuromuscular junction. That's exactly the same picture. The acetylcholine, when the signal reaches here in the synaptic, that's the action potential. When it reaches here, the acetylcholine gets released and they bind with the membrane bound receptors on the postsynaptic cell. So this postsynaptic cell receptor will decide whether they will be excited or inhibited based on their characteristics, based on their interaction. And then once this ends, these neurotransmitters get recycled. That's what I wanted to tell you one more time. And this is very interesting also, because if you go back here, let's say there is there's another neuron in your brain, they release serotonin. I'll just give you an example to understand. They release a neurotransmitter called serotonin. Okay, serotonin, what they do, they bind to the receptor and makes you happy. You have a better like feeling. You have a happier feeling. You're in utopia, like you're feeling better because of the serotonin. Now, but remember, this binding is not forever. It's not an infinite time. There is enzyme, they're going to degrade the binding. And when they degrade, that serotonin will go back exactly like acetylcholine. Then you'll be sad again. So your mood is changing from happiness to, and that is exactly what happened in the brain. Your mood is changing. So as soon as you get the grade, you got an A, you release tons of serotonin, you're happy, made your day, and do whatever. But as soon as the grade is released, you did not get the one that you thought you're going to get, and then you're sad, meaning your serotonin probably is not released, okay, or any inhibitory one is got released. So they discovered the drug based on that. See that there's a drug called Prozac. It makes people happy. So when people are depressed, they take this, the doctor prescribes this. What they do, they literally serotonin reuptake inhibitor. Actually, it's called selective serotonin reactive inhibitor, meaning SSRI drug. So basically what the drug does, they don't allow that serotonin to go back. Remember, the serotonin is coming out and they go back to the drug, they will stop the serotonin. They will keep the serotonin there in the junction for a longer time. So the person will be happy longer time. That's exactly how it works. Okay? I hope you understand the entire concept of the signal, the neurotransmitters binding with the receptor, presynaptic and postsynaptic cells. Now, final part of this chapter is few information regarding neuromodulators and how they work. So what neuromodulators are, without going very detailed, because you do not need extreme detail about this particular area. Neuromodulators are basically when we say that presynaptic cell is releasing neurotransmitter, they're not releasing one variety, they may release multiple varieties. So obviously follow the name. Neuromodulators, what they do, they alter the rate of neurotransmitter release. So they are basically helpers. Imagine they're like a sous chef. So chef is the neurotransmitter, and all the sous chefs, those are neuromodulators. So they basically, they're mostly neuropeptides. They can also bind to the receptor. They can also do the same thing. It's like sous chef. Sous chef actually does exactly the same thing what chef does, but they're considered like help. Same thing here, neuromodulators. They're helping neurotransmitter to achieve their goal. Okay, there are multiple of them. You just remember one class, one variety for your test purpose, opioids. Opioids is a very common one. That's I'm going to say that because these are morphine categories. So what they do, their primary function, they block the pain receptors so that you don't feel pain. You know, morphine does not give you pain, so morphine reduces the pain. So the opioids are variety of types. These 4 types, memorize their name: enkephalins, endorphins, endomorphins, and dynorphins. They have a little bit different in mechanism. Again, all this detail you absolutely do not need at all. So what you need to do in this particular slide, you need to know what is neuromodulators and what are opioids, how they work, like their primary function is to block the pain receptors, and how many types are they? 4 types: enkephalins, endorphins, endomorphins, and dynorphins. That's good enough for information here. But whether it is neurotransmitters or it is neuromodulators, one common theme is they're going to bind to the receptors. One more time. So whether it is neurotransmitters or neuromodulators, we're learning throughout the lecture that they're going to bind to the receptors. Now, when they bind, where is the receptor and what happened then? That's what we're going to study right here. So they fall into directly 3 categories, very straightforward. One category is once they bind, let's say acetylcholine, that's our neurotransmitters. Once the neuromodulators does the same job. Once they bind to the receptor, boom, the ion channels open. Receptor in this case is the, let's say, related, directly attached to the gated channel. So gated channel opens, ion goes in. That means signal is transferred. So basically this is called direct effect. Direct effect means directly they are influencing the ion. So direct effect, or sometimes they also known as ionotropic effect, directly entering, the ions are entering. So directly ions are entering. Just remember these 2. So that means direct effect means ionotropic effect. Indirect means, let's say the neurotransmitter bind to the receptor. So once they bind, they become active. Then that receptor, you don't need to know the detail of this. The receptor goes through a lot of cascade of reaction, lots of mechanism of reaction. So I'll give you an example. So you go to a room, you open the door handle, boom, open it directly. So you press the door handle, you open the door directly. Some other type of, you press a button that send the electrical signal somewhere, some down the line. They send the signal to the, let's say, camera. Somebody's in the central, monitoring area, let's say they're looking at the camera. And let's say you have a so-called ring video cam, bell, and then they look at your camera and then say, okay, you know what, I'm going to allow this person. So they press a button on the iPhone and that goes signal back to the lock unhinge, unopen, you open the door, or the door open automatically for you. That's like an indirect effect. So the first one, you touch the door handle, boom, you go in. That's this direct effect. The second one that I just explained, so they go through a lot of metabolism. That's why it's called indirect or metabotropic effect, because it's a cascade of reaction, a lot of metabolic reaction. And then finally the ion channel is opening. That's called metabotropic effect. And finally, that one that is some cases the receptors are, there is no receptor because they can easily directly move. Their receptor is considered is like a cytosolic, meaning they can directly move. Some neurotransmitter or neuromodulator, they can directly move through the membrane, such as nitric oxide or carbon monoxide. They can directly move through the membrane and promote the second messenger reaction there. So they don't need to press that button and wait for the signal. They can go inside and promote their own cascade of reaction. That's no separate name, but that's an effect directly inside without the receptors. Okay, so indirect effect, this is indirect effect via intracellular enzyme, meaning they are not doing anything to the receptors binding G proteins. They're directly inside. So they're causing indirect effect, but they're through the intracellular enzyme, which cause second messenger pathway. So the difference between this is here, they have a receptors which attach to the G protein and then they do this. Here, they don't need that. They can go directly and affect this second messenger pathway. So they don't need to press the button and you want to do iPhone. Here, I have, this is my door. I have my iPhone. I can press a button, then I can open the door indirectly. Okay, I don't have to your permission. So this one, I need your permission to get in because you have a receptor. So that's the difference between these two. And this is direct. You open the door by turning up the handle. So that's basically this entire chapter. The last slide I found somewhere that basically described the neuron structure, just to keep you memorize one more time. So this particular video, you should focus firstly, first you should watch that video and read the book as usual, take a note on the PowerPoint slide that we have, and pay attention, divide into two parts. First part, you just learn what is action potential. So you need to know sodium channel, you need to know potassium channels, then you need to know mainly local current, which cause a graded potential, and which then, if they reach to the threshold potential, then they can go up to action potential. So you need to know the definition of graded potential, action potential, threshold potential. Then you need to know transmembrane potential, that is, or resting membrane potential, which is negative 70 millivolt for neuron. Then you need to know all the gate. What are the gated? Chemically regulated gate, mechanically regulated gate, and voltage regulated gate. These are the three types of gates available. Then we know how action potential happens. So action potential happens, and then there are two kinds of propagation. One is saltatory, one is continuous. Continuous is slow, unmyelinated neuron. They're like a local train. Continuous and express train, they don't stop everywhere. They just stop at node of RANVA. So they are express, myelinated neuron. Then according to the size of the neuron, if it's the thickest, it will take the transmission of the signal fastest. Type A fiber, type B fiber, type C fiber. So know those three types. Then you talk about neurotransmitters. Neurotransmitters could be excitatory, neurotransmitters could be inhibitory, based on which receptor they bind. And at one page, we talked about some neurotransmitters such as acetylcholine, GABA, gamma-aminobutyric acid, norepinephrine, epinephrine, dopamine, serotonin. These few good enough. And then you have neuromodulators. The definition of neuromodulators, as I explained you, is like nut shape, is a shoe shape. And then one variety is opioids. There are 4 different classes. You should know those 4 classes. And then their main job is to blocking, their overall job is to block the pain receptors so that people does not feel pain. And then finally, you study their effect, either direct or indirect. Direct means ionotropic effect. Indirect, when via G protein, we call them metabotropic effect. And when they do directly, they don't have any special name. So they just call indirect effect via intracellular pathway. Okay, thank you.